A typical angiosperm is a bilobed with each lobe having two theca. That is, they are dithecous. Figure 1.2 shows about this diagram, about these lines. A typical angiosperm anther is a bilobed. You can see that anther is a bilobed, two lobes, and each lobe has a two theca. You can see in this picture. You can see in this picture that bilobed and it has a two theca. Okay, and they are dithecous. Often a longitudinal groove runs lengthwise separating the theca. Often the longitudinal groove runs lengthwise and it's separating the thecas. Okay. Let us understand the various types of tissues and their organization in the transverse section of an anther. The bilobed nature of an anther is very distinct in the transverse section of the anther. This is the bilobed nature of anther. It is a very distinct when we cut in the transverse section. This is the transverse section. Okay. The anther is a four-sided tetragonal structure consisting of four microsporangia located at the corners, two in each lobes. The anther is a four-sided tetragonal structure consisting of the four microsporangia 1, 2, 3, 4. Okay, and are located at the corner, two in each lobe, 1, 2, 1, 2, two in each lobe. This is the anther, this is the filament. Next, the microsporangia develops further and becomes pollen sacs. Okay, the microsporangia that I shown now, that develops further and becomes pollen sacs. Okay, they extend longitudinally all through the length of an anther and are packed with pollen grains. Pollen sacs extend longitudinally along the length of anther and pollen grains are packed in that. Structure of microsporangium. In the transverse section, a typical microsporangium appears near circular in outline. It is generally surrounded by the four walls: the epidermis, endothelium, middle layer, and tapetum. Okay. In the transverse section of the microsporangium, it is circular in outline. It is circular in outline. I will show the picture. You can see that it is circular in outline, the transverse section. Circular in outline. And it consists of the four layers. That is the epidermis, endothelium, and then the middle layer, and then the tapetum. Okay. These are the four layers. First, you have to remember these words. Four layers. Say E is two layers starts from the letter E and E. Epidermis and then the endothelium, and then the middle layer and tapetum. This is the tapetum. This is the sporogenous tissue. It's a connective tissue. Okay. You can see in this picture also. This is the epidermis and endothelium, epidermis, next layer is the endothelium, middle layer is the middle layer, microspore mother cells, and this is the tapetum. Okay. And functions of these layers are, next we can learn the functions. The outer three layers perform the function of protection and help in the dehiscence of anther to release the pollen. Okay, the outer three layers means epidermis, endothelium, middle layer perform the functions of function of protection and help in the dehiscence of anther to release the pollen. Okay, the innermost layer, that is the tapetum, It nourishes the developing pollen grain. It nourishes the pollen grains when they are developing. Cells of the tapetum possesses the dense cytoplasm and generally have a more than one nucleus. Tapetal cell possesses the dense cytoplasm and it has more than one nucleus. Can you think how tapetal cell would binucleate? Can you think of how tapetal cells become binucleate? Okay, you have to find the answer for this question. When the anther is young, a group of compactly arranged homogeneous cells called as the sporogenous tissue occupy the center of each microsporangium. When anther is young, a group of compactly arranged homogeneous cells, okay, not the heterogeneous cells, called as a sporogenous tissue. Homogeneous cells when which occupy at the center when anther is young is called as a sporogenous tissue. It occupies the center of each microsporangium. Next one is the microsporogenesis. As the anther develops, the cells of the microsporogenous tissue undergoes meiotic division to form the microspore tetroid. What would be the ploidy of the cells of the tetroid? When the anther develops, sporogenous initially forms sporogenous tissue from the homogeneous cells undergo meiotic division to form the microspore tetroids. Next is the question, what would be the ploidy of the cells in the tetroid? I will answer in the last video of this chapter for all the questions. These are the diagrammatic views. These are the diagrammatic views that we can see. Already I explained these two diagrams and this is the third diagram. These are the pollen grains. These are the pollen grains. A diagram is the transfer section of the young anther. B is the enlarged view of the microsporangium showing wall layers. Third is the immature dehiscence anther. Okay. Next one is the as each cells of the sporogenous tissue is capable of giving rise to a microspore tetrad, each sporogenous tissue is capable of giving rise to the microspore tetrad. Each one is a potential pollen or the microspore mother cell. Okay, each one develops into the potential pollen or the MMC microspore mother cell. The process of formation of microspore from a pollen mother cell through meiosis is called microsporogenesis. Okay, the process of formation of microspore from a pollen mother cell through the meiosis is called microsporogenesis. It can be asked for the second page examination. What is microsporogenesis? You have to define microsporogenesis like this. The process of formation of microspore from pollen mother cell through meiosis is called microsporogenesis. The microspores as they are formed are arranged in a cluster of 4 cells. Okay, they are arranged in a cluster of 4 cells. I will show in the diagram that are the microspore tetrads. Okay, I will show only the diagram. Okay, see, this is the microspore tetrad. It forms the microspore tetrad, no? This is the diagrammatic view of that. Then the microspore tetrad, as the anther mature and dehydrate, the microspore dissociate from each other and develop into the pollen grain. When the anther mature and it dehydrate, the microspore dissociated from each others and develops into the pollen grains. Inside each microsporangium, several thousand of microspores or the pollen grains are formed that are released with the dehiscence of anther. Inside each microsporangium, there are several thousand of micropollen grains. It also called as the microspores are formed and are released with the dehiscence of anther.